Welcome back to End is the Future, a podcast that brings together great minds to address the opportunities and challenges of making businesses both sustainable and profitable. I am Ilham Kadri, and I'm delighted to be your host today. If you are as passionate about sustainable business as I am, let's explore how End is the Future together. Today, I'm thrilled to be speaking with Professor Katalin Karikó, a dear friend and a brilliant scientist. Professor Karikó's research led to the creation of the messenger RNA Covid-19 vaccines, which has saved so many lives and helped curb the impacts of the pandemic. Her groundbreaking contribution was to use chemistry to modify the mRNA so that it wouldn't be rejected by the immune system. And she has quite literally changed the world. I had the great pleasure of meeting Katie when she was chosen as the winner of the Nobel Prize for her incredible achievements in the field of chemistry, and it was so well deserved. So thank you, Katie, so much for joining me today.
Thank you, Ilham, for inviting me. It was such a wonderful celebration in March at Solvay. Me and my daughter will remember for that forever.
And we will remember you forever, I'll tell you, our employees, our audience, our key stakeholders. So, Katie, most people know about your scientific achievement now, but many people may not know your backstory and what led you on the path to your career. Can you tell us a bit about your upbringing in Hungary and what sparked your passion for science and chemistry?
I grew up in a small town. 10,000 people lived there in Hungary, in the middle of a plain. And it was a very simple household. My father was butcher and my mother was bookkeeper.
Yeah.
And also, of course, she was running the house. And I had an older sister. She's 3 years older than me, and she was very good in school. And so when I entered school, the teachers expected that I would be a good student. Well, and our parents, of course, encouraged us to study well. And I don't remember, but I was told that when I was even 5 years old, I watched my father opening a pig because he was butcher, and I want to see what is inside. My sister, who was, as I mentioned, was older, she ran away and she was not interested. Actually, eventually she ended up, became an economist, get a PhD in economics, but that, she was not interested. What is inside the pig? But I don't remember, but I was curious. And then the teachers were excellent in elementary school and high school. And so I just stayed curious and wanted to learn more, and successfully could enter to the university, which was a very big deal. It was very, very difficult to get to the University of Szeged. And there I went there, Szeged, because there is a biological research center there. And when I was a student, I started to go there and I tried to do some kind of work. And first I ended up in the lipid lab, which for most of the people were considered very boring thing. But because at that time, I am talking about the 70s, mid-70s, actually genetics was the most important topic.
Mm-hmm. Yeah.
It was kind of a boring thing. But this is where it happened that we formulated and put plasmid to lipid wrapping and delivered to the cells. And it was in the end of the 70s. And it was, voila, we delivered this molecule inside the cell, and then the cell expressed the encoded protein. And it was, for me, was very important because I keep thinking about this first experiment. And that's why I suggest for that it doesn't matter what you do, you just have to make your hands wet, try something. And of course, how I started to work in 1978 with RNA, it was just by chance. The head of the RNA team walked in one day to the lipid team, and then my supervisor at the lipid team said that Kati wants to make a PhD. And then the next time I already went to the RNA team. So everything was by chance. I did not come up with the idea to work with RNA. But of course, when I started to work there, I found it very exciting and interesting molecule. And so I like to work with RNA. We made chemically, enzymatically short RNA molecules.
Yeah. And it's amazing, Katie, you talk about curiosity, right? And the importance of being curious, which is important for scientists, for our researchers in the industry these days, but also for leadership. And I remember you told me when we met a few months ago that experiments do not fail, only our expectations fail, which since then I've been stealing it from you, which was so inspiring. And one thing which really struck me, and you are in Hungary today, right? That's what you told me, right? Visiting your sister. I remember when you were speaking about you leaving Hungary to go to the US, and that back then you could only take $100 out of the country, or euros. I don't know what was it at that time. So you had to get creative. Can you tell us more about that?
Yes. So in 1985, when you get a job offer from the Western country, only your family member were allowed $50 to take. So my daughter, who was 2.5 years old, and my husband could take $50 with, from the bank. But as me, getting the job offer, they said, I have to get money from whoever offered. Of course, why the American university would pay me in advance? Of course, this wouldn't happen. And so we sold our Russian-made car, and then we had a student from Arab countries at the medical school, and then they had currency that I could exchange. And finally, I think it was £800, which I could manage to work. And we had to hide it because it was illegal. And finally, I opened up the back of my daughter had a teddy bear get from my mother. I opened up the bag, and then I put the money wrapped up. At the airport, we were watching my daughter and with the teddy bear. And actually, she was the smuggler because nobody found and nobody was looking for that extra currency.
I'm curious. Do you have still the teddy bear somewhere? Do you have it?
Yes.
Yeah. Okay.
He was still there.
Yes, I would love to see the teddy bear. Now let's talk about your scientific breakthrough, actually, the messenger RNA technology. Sometimes timing can be everything, right, Katie? And I imagine we are in January 2020, right? We all lived this, right? Painfully in our ways, from China to Europe to the US. And it just happened to be the case that you were already working on, I think, a flu vaccine, if I remember well. And then the coronavirus hits and everything came together quickly. How come a vaccine can be developed in 9 months, Katie, where, people were telling us you need years. So can you tell us more about that?
Of course. So the development of messenger RNA for therapeutic application, for vaccine application, it was decades, several decades. And the scientists painstakingly learned more and more and improved the RNA quality on a way that more protein could be produced from the RNA. It made it, we learned how to clean it, how to make it pure. We also learned what kind of material we can wrap it up so it can deliver easily, because this RNA had to be protected when it is injected to the body. So that was also a lot of development there. If we don't have this technology, we had it 20 years, but we wouldn't have, then everybody would wait to start to make a vaccine, to receive a package from Wuhan, which would contain some kind of viral material. We didn't need that because we just needed the information. So when information became public in 2020, January 12th, everybody could read it and they could proceed without getting material in their hands, because everybody could order or synthesize the genes and started with their procedure, which was, their favorite one, whether they made protein, whether made DNA or RNA-based vaccines, or incorporated some kind of viruses. So different kind of viral vaccine technology existed at that point, and everybody could proceed it because they already had the information, and that was sufficient. we already learn each other. We learn how to work together. And by the end of 2019, we were ready to proceed to the human trial with, the influenza, against the influenza. But of course, the technology is so simple and so straightforward. You just have to change the template from which you copy the RNA, so that changing the template was efficient to switch from influenza to the new pandemic candidate, this coronavirus. And so the timing was perfect. And if it would happen earlier, of course, we would have much more difficulties. But by 2020, we could act. All of the animal experiments were done already with the vaccine, which the formulation was identical, which was, the composition of the nucleic acid was identical from the 4 basic nucleotides. What was the capanol? Everything was identical, which we already tested out for influenza in animals, even in monkeys. So it was very safe.
Yeah, so it's amazing, you were working and you have the framework. And as soon as probably you had the sequencing rights of this coronavirus, you could just mimic and bring it to the influenza framework. But then this mRNA technology, Katie, can be applied to many other diseases. Can we dream about curing cancer, malaria, tuberculosis, right? What is the next big thing for mRNA?
You might be aware of that many of the vaccines are, in the advanced stage. Actually, in Moderna already has a phase 3 trial. The CMV and RSV viruses already injected subject at phase 3 trial. And they also have, HIV and, of course, BioNTech also, the influenza and others. And in human trial. And so in several is in human trial, and we also can see some is advancing towards human trial, like herpes simplex 2, for example. And not just the viruses, but also for parasites like malaria you mentioned.
Mm-hmm.
Already a couple of years ago, we have seen already data, animal studies data. Mm-hmm. The difficulties, of course, there is that the organism, the microbe has much more genome, and you have to identify what you had to fight, what the RNA should code to have a very effective vaccines. And this is ongoing. And also for tuberculosis, which is an intracellular bacteria, and identifying what are the critical elements that we should code by the mRNA to get a very effective vaccine. So these are ongoing, but you could see that some vaccines we already have tried to replace it with a cheaper vaccine, because the RNA is cheap, and so it is much of the equalizer. Many drugs which were unaffordable for people with the mRNA-based materials, not just for vaccine, but for passive immunization or therapeutic protein. Those are very expensive, but if you deliver the messenger RNA code for the protein, it will be affordable for everybody.
So before we switch gears to your journey, right? And some of the attributes of I'm keen to share with our audience those attributes. I talked about curiosity, risk taking. There are more in the journeys of Katie. What was it like the day you received the vaccine? That was the result of your research, Katie.
It was special. I thought about that. I am, I know so much about what is inside that vial, because we work very hard optimizing different elements of the RNA and the cap structure and other things, and of course it was just so unbelievable that it happened. We worked on something, and then I am here. I am, and I am benefiting by protecting myself, and so. And those people who were around, healthcare workers waiting for another room for the vaccine, and they're cheering us and clapping and being so happy. They could finally can breathe and go home, with knowing that they are not infecting their families or something, is because they are the real heroes. We all know, because they went day after day for a whole year and treated patients and risking their lives.
Yeah, absolutely, Katie, and it's good to pause and applaud, recognize our invisible heroes. You're right, but yet I think there is another attribute, which is humility from your end. But we all cheer you, by the way. So and coming back to your story, I mean, it's an incredible story of resilience and determination. You faced throughout the journey many challenges, right, in your career, but I know you persevered and went to making on of the most important scientific discoveries that has and will continue to have such profound impact on us, on healthcare. Can you tell us a bit of the setbacks and explain to our listener what was like, and how did you stay resilient and never give up?
Yes, actually, the Sunday Le Monde has inside an article about, I wouldn't be here if, and I followed, if I wouldn't be terminated in my position several times. So with that one, I want to say that it's never the decision what other make, that you have to leave your position, but what you will do. So the power is in your hands. And instead of, complaining and saying that why I am terminated, because probably I can look around and find other people who might, produce less. And so I suggest everybody do not spend any time, agonizing, licking their wound that why they are terminated. They have to focus all of their energy on what next and what to do now. And with full of optimism that they are able to do that. And so I was terminated in my position in Hungary, and I never wanted to leave and go to the United States. I was, I was sleepless night when I realized that I cannot stay in Europe, where I applied for 3 positions, and everybody wanted me to bring them stipend or some kind of scholarship with me and come to the lab. And I had to go to so far away that I never wanted to go there. And so sometimes, you are in a position and then you just have to do it. And then also, even in the United States, I lost my position and simply just I was kicked out. I really, Ilham, I said loudly that I am in the United States of America. Where else, if not here, I can do what I wanted to do, and then I just encourage myself that doesn't matter, that I am not faculty anymore. And again, when, finally I was ready to leave the university, we had the patent and we established it through Weissman with my colleague, a company. We had our world patent, but finally the university decided not to give it us. So again, that was what more painful. And again, when I was 58 years old and finally, shown the door that, I was not successfully getting money, grant money. I understand, because there in the United States, if you don't have money, don't have space. I don't have any grudge against any of these colleagues, they were in a position and then they follow the rules. So again, I wouldn't go. But again, at 58, I was said that, time to go. And then I decided to go to biotech. Again, at a company that other people wouldn't even know about, because they didn't have a website.
Absolutely.
And just have the faith that, okay, I will go there and start. And we all work together. And it was a very wonderful time.
Yeah, and amazing stories again. A few months ago, Katie, I had Nobel Prize winners Stephen Chu and Ben Feringa. You met at Maison Ernest Solvay, our founder home, right, in Brussels, who were on the jury, by the way, to choose you as the winner of the Solvay Prize. And we had such fascinating discussion about failure in scientific research. And I must say, in our modern world, failure is a bad word, while in science and with my background as well, I mean, it's something you are not ashamed about, right? And I remember Stephen Chu mentioning that in scientific research, you had to go from failure to failure to failure with continued enthusiasm, which I thought was a great way of putting it. So I think failure in science and what it leads, but also in your journey as a professional, as you said, right? But it looks like you didn't get frustrated and you were just focusing on your journey, right? And just pursue your dreams, right, Katie?
You have to see the progress, and that was important. I could see that the protein translation is getting better, the RNA is more effective, and I could see it. Of course, you couldn't see that I am advancing because many times I said, my career was like this Hungarian Csárdás, there is not forward step. It is always left and right and left and right, and nothing forward, but things happening. So that's what I'm saying. Also for these young ones that don't try to, please somebody, the supervisor, and don't feel that you work for the company or the university. No, you have to focus on that. You try to understand the part of nature, and if you would, this would be your primary goal, not your advancement in career. Then you wouldn't be disappointed if you could see somebody's publishing what you are working on, because you say, okay, they help me in to understand better.
Yeah. Well, what's another lesson? It's about inside out, right? And how to make it, your journey. So, I mean, your life has completely changed, Katie, right? I mean, you've gotten more award than we can probably count by now, including, I'm proud to say, the Nobel Prize, right? But what has it been like adjusting to this almost overnight fame? Are you enjoying it? Do you miss the lab? Do you miss, the bench? Where are you?
Yes, most of the thing I miss is that when I can see a paper and I can see the title and I can skip a little bit to the abstract, and I have the feeling that I just do not have time to read, and I think that is something important. So I decided that next year, 2023, I go back to science. I have to do this because there are so many things we need to solve, and then I accumulated so much knowledge that I can have in. And listen, Ilham, I never dream about that my name will be put forward. I never wanted the personal recognition. Many times I thought that, 100 years from now, nobody will remember who we are. It's not important, focusing on something, some project which is really makes a difference in somebody's life, treat some disease. That's what we have to focus on. And then now that I became known and in the spotlight, I try to find some voice. What should I do? And the one thing I try to, help with the many reporters to educate. And now today with you to educate the public, because I realized that some resistance against the vaccines is coming, that we were not doing a good job to educate the public. And we learned that they can talk about mRNA, they talk about PCR and other things they have some idea about. And we should rely on that. They should know more and understand, because if they feel only the unknown. And if they would understand better, then they would, embrace it, and they would understand and champion the scientists. I really was shocked when I asked people, that to name scientists, and they name people who are not with us anymore, and they cannot name a contemporary person who is a scientist. They are taking medicine which saving their lives, and then they don't know. They know the football player, the actress's name. But what is the person who just discovered and come up with this checkpoint inhibitor, James Allison, and then saved my life, and then I don't even know their name. I don't even ask for it. So that's who's responsible. So I try to champion the science and champion the women. So I also try to have that more attention will be on women and their importance, because we are differently thinking, and we are multitasking, and we are just as valuable for science. And there are so many things which people are asking me, how I could work and be a parent and raising a child and science. And you never ask this in a fellow male scientist.
Absolutely. And it's so true. And I bet you will be remembered in 100 years. I think you are becoming, whether I know you didn't want it and you don't wake up every morning thinking you are a role model, but you are an incredible role model for so many girls and women in science, technology, engineering, and math. And this is so important considering you desperately need more women to take these paths. When I was a young girl, my role model was Marie Curie. I wanted to be Marie Curie, another one, inventing things. I didn't know how to do it because just loved STEM, and she was the only woman I knew of. So so tell me, I'm curious, who did you have role models? Who are your role models? Who inspired you? And what are your tools? I think you start talking about diversity, equality, and inclusion in STEM careers.
I mean, when I was in school, I remember that my high school biology teacher, whatever we taught, he already knew so much about it, and I wanted to be like him, to know everything about everything. And of course, my parents who work very hard, and then also that I learned from them, so that working hard is part of our life, and then learning constantly that when I went to the university, others could speak English. I 18, I couldn't say. You can hear the heavy accent. I couldn't speak anything, and then others already had chemistry, and then what meant that I could see the enormous progress I have to catch up. Meanwhile, others maybe laid back, and more complacency was there because they already so much advanced, and by the time they woke up, they realized maybe that I already took the English exam and took, I advanced, and I could not ever lay back and think, because more I learned, more I realized that I have knowledge so little. I have to learn that and that, and many times I was just shaken that, oh my God, I think I'm a biologist, my 10 years doing something, and I didn't know that, and then spent the whole weekend just studying. And it was fun. That's what I have to emphasize. For the little girls also, because people said, oh, you struggle. Come on. I was so happy in the lab. I was in the chair, running the sample, figuring out things. And it was fun. It was, yes, the motion and send me away. Yeah, that it was, but always come up with a solution, what to do next. And then get back to where I am in full control. And it is fun to solving problems, and even you ask the questions, you try to find the answer, you do an experiment, and more technology, the techniques you learn, more knowledge you have, that it's just a delight to work, and we get paid for it. So it is. I want to see that part, not from what right now can be seen outside, that somebody's working day and night. And I went there because I wanted nowhere else to be. Not that I would be promoted. I already was promoted. I went there because I came up with a new idea what to do. And that's why I went the weekends. Nobody forced me. Nobody. If I don't go, that's fine. I wouldn't get kicked out from there. It is just, I want it because that's where I have fun and enjoyed, and I had a wonderful husband who did not complain about, and who fixed up the equipment and so on. So this is a fun thing to do, and you spend all your life working. So why not to spend with something that you are enjoying?
I mean, I know that a lot of young scientists will tune to this podcast to listen to you. So is there any other advices would you give to these scientists, right, who are starting out?
Many young scientists get disappointed because they are comparing themselves with others, and they can see and conclude that, oh, they are better, and these others are, maybe getting more advancement and promotion and more money and whatnot. And they get disappointed and they just leave because, and I'm telling, not pay attention on those things you cannot change. And you cannot change some people advancing and they are already doctorate or whatnot, and I can feel I am 40 years old, I am just demoted. Doesn't matter. You are getting better and better if you keep learning, understanding, doing more experiment. And of course, you have to enjoy. If you do not enjoy in the laboratory work and thinking about reading about science, then you have to go to another profession. And if you like to be in the spotlight, you have to be actor or actress or some other performing art or, to do something. When you have criticism, you have to listen. Do not take it personal. Somebody already spent time to tell certain things to you. Listen, think about what you should do. I grant when I was rejected, my grant is not saying that, oh, those people are stupid, do not understand. Even if I would say that, okay, they didn't understand because I did not write well or something. So focus on that. Okay, how I can make it better. Actually, I just get an honorary doctorate. And when I thanks to all of the people who helped on my journey, I also said, I have all of those people who try to make my life more difficult.
Absolutely.
Without that, again, I wouldn't be here. They make me, more resilient.
Yeah.
You will learn from everything. If somebody treats you badly, you learn from it that you won't do this to another person. You learn always, whether it's good thing or bad, but learning is part of it. That's what I can tell the young ones.
It's and I always say feedback is a gift, so take it as a gift, right? And it's so profound and resonates with me and with us. Well, thank you so much for joining me, Katie. Not only you are a brilliant scientist, but frankly, and more importantly, you are a beautiful soul, and I got to see you even in private setting, and such an incredible inspiration for many young women, and not as young women like me, by the way. So I have Marie Curie and Katalin Karikó by now as a role model. So this was a fascinating conversation, and I can't wait to see what's next for you, Katie. Thank you.
Oh, thank you very much.
Thank you so much for joining us today. For more information or to listen to other episodes, visit our website. And if you like these podcasts, don't forget to subscribe. Until next time, be safe.